Now we will see the artificial seeds. Artificial seeds are synseeds or synthetic seeds. Artificially they are produced. They are not natural seeds produced from the plant as it is. From the fruit they are not produced. Artificial seeds are produced from any cell of the plant culturing in the culture medium. When any cell of the plant culture in the culture medium, in the plant tissue culture, that will form callus. The callus will develop into embryoid. The embryoid can be used for the production of synseeds or synthetic seeds or artificial seeds. The cell, when one cell is taken from any part of the plant that is grown in the culture medium under aseptic condition, so a cell mass will be produced. That cell mass contains dense cytoplasm, large nucleus, starch grains, proteins, and oil. And that is the product of the cell. One cell gives rise to a cell mass with dense amount of cytoplasm, large nucleus, starch grains, proteins, and oil. And that cell mass is converted into callus. From the callus, some embryoid somatic embryo will be produced. Those embryoid will be coated with agar cell and sodium alginate. That's the inert materials which will be coated, applied on the surface of the embryoid to get synseeds. So that the end product is synseeds. Synseeds or synthetic seeds and artificial seeds are produced like this. This is the protocol, the method for the production of artificial seed production. Number one, a single cell selected from any part of the plant. First method. Second method, the single cell is taken in the culture medium, in the aseptic condition, in the culture vessel containing culture medium, under aseptic condition, that cell is grown, that forms cell mass. A parenchymatous, undifferentiated mass of parenchyma cells that contain dense cytoplasm, large nucleus, starch grains, proteins, and oils. That slowly will converted into callus. Callus, as I told you, it is a undifferentiated mass of parenchyma cell. The callus, by hormonal concentration, if you supply hormones, plant hormones to the add with the nutrient medium, to the nutrient medium, that converted into somatic embryoid. Somatic embryo will be produced, that is called embryoid or somatic embryoid or somaclonal embryo, that is directly produced from the callus. When the somatic embryos are taken and they are coated with agarose gel and sodium alginate, that is converted into synthetic seeds. That is the artificial seed production from the embryoid. Now you will see advantages of the artificial seed. First advantage, that can be produced at a very low cost. That's the first advantage. Second, it's an easy method to produce genetically engineered plants. Genetically engineered plants are plants which are modified plants, genetically modified plants, and they contain foreign gene from another plant will be inserted, that become transgenic plants. This is the easiest method to produce genetically engineered plants with desirable characters. Third one, it is easy to test the genotype of the plants. Genotype means the genetic constituents of the plant. The genotype can be easily tested with the help of the synseeds. Fourth one, they can be stored for a long time under cryopreservation. cryopreservation means it is a method of preserving some of the plant product under minus 196 degrees C in liquid nitrogen. So cryopreservation, it is a novel method. It is a special method by which the important parts of the plants, like seeds of a very rare plants, pollen grains of the very rare plants, can be preserved for a long time, very long time, under minus 196 degrees C in liquid nitrogen. That is the cryopreservation. So these syntheses can be stored for a long time under cryopreservation method. And the synthese will produce identical plants. All the plants will be similar in structure. The plants which are produced from the synthesis will be alike, genetically identical plants. Growth is faster because dormancy period is very less. What is the dormancy period? The resting period of the seeds are called dormancy period. Here the dormancy period is much reduced, so growth will be very fast. The growth will be very, very fast with a short life cycle. So these are all some of the advantages of artificial seeds. Can be produced at low cost, easy method to produce genetically engineered plants with desirable traits, easy to test the genotype of the plants, can be stored very long time by cryopreservation method, and produce identical plants, and the growth is faster because the dormancy period is very short. So that is about artificial seeds, Now come to protocol for virus-free meristem culture. How to produce virus-free plants? It is not possible to produce virus-free plants very easily. This is the only method, apical meristem culture, shoot apical meristem culture, shoot tip apical meristem culture, shoot tip meristem culture. That is the only method by which virus-free plants can be produced. Other parts of the plants are easily affected by the virus. There are many chemicals which will destroy fungi, bacteria, which may cause disease to the plants, but virus cannot be destroyed by using any type of chemicals. But the tip of the shoot is not affected by virus, so the meristem which is present at the tip of the shoot is isolated, separated, and grow in the culture medium under aseptic condition. It is possible to produce virus-free plants. So this is the protocol for virus-free meristem culture. First, apical meristem tip should be cut from any part of the plant. The tip of the shoot is removed, and that is with leaf primordia plant is taken as the explant. Apical meristem tip with some of the leaf primordia. Leaf primordia means the very initial stage of the leaf, that is the leaf primordia, should be taken. That is the explant. Already I told you the part of the plant is used for plant tissue culture is considered as the explant. Second, in vitro culture, in 10 ml of solid MS medium supplemented with growth hormones is provided to the plant, to the culture it is provided. So in the in vitro culture medium, the in vitro culture medium we have taken is MS medium. Along with MS medium, growth hormones should be supplied. Culture are maintained at 24 plus 1 degree C or 24 minus 1 degree C that is the incubation period, and that is maintained in the dark room for 3 days, followed by natural illumination of 2400 lakhs. If you follow these conditions, naturally that plant material which we have taken will produce plantlets which will be developed from the meristem tip culture after organogenesis. First organogenesis will be taking place. Organogenesis means formation of root, formation of stem is the organogenesis. After that, from that the organogenesis from the organs of the callus, plantlets will be produced. Plantlets are very minute and microscopic. That will be minute, not microscopic. They will be very small, soft and delicate. That is transferred to hardening process under glass room, under greenhouse room. These plantlets should be grown in order to adjust with the natural environmental conditions. So step by step we have to give hardening. Then after that the plantlets will be ready in order to grow in the field condition. That will be transferred to the field condition. This protocol for virus-free meristem culture. That's all.